Okay, so the last part on this topic is actually on the proteomics part. So first and foremost, let's have a look at what is proteome. So as you may know, because since you have learned about genome, so protein is basically something that complement to a genome. So proteome is defined as a full set of proteins comprising the structural, metabolic, and any regulatory machinery of a cell, tissue, or organism. And unlike genome, whereby you only have a genome of an organism, which means that the whole genome, basically there's only one genome comprising of one organism. But in terms of proteome, different parts of the organism is considered as one proteome. For example, if it is from a human, okay, the ear proteome, the nose proteome, and so forth. Okay, right. So now, as you have an omics at the end, so proteomics means it is a study of a protein. So you could study in terms of qualitative and also quantitatively, and you could also do a comparison under different conditions, and it could be directed to unravel biological processes because we know that protein is involved in biological processes. And proteomics is one aspect of systems biology. So basically, systems biology, I define it as simple as I can, a case study of mechanisms underlying complex biological processes as integral systems of many diverse interacting components. And we know that systems biology is actually very big. So one aspect of functional genomics and differential gene expression is proteomics, okay, because the expression is regulated at multiple levels, and because at protein there are a lot of things being involved, and there's also a lot of protein isomers. Now we know that protein, just like gene, could be regulated. So regulation is in terms of starts from the transcription, from the RNA splicing, okay, up until translation efficiency, post-translational modification (PTM), protein-protein association, degradation, and also we know that even though for genome, okay, and probably Dr. Fatah would have covered it, you could get more than 30,000 genomes being identified or unraveled from the human genome, but it could basically only comes up to about 1 to the power of 5 or to the power of 6. That's it, okay, in terms of protein. So the number would be reduced, okay, once it is being translated to protein. Alright, but we know that protein is actually a bit more complex, so it would be inherently more challenging than genomic or transcriptomics. And you could see probably just by side-by-side comparison of this. So on your left would be the nucleic acid genomics, or either transcriptomic, and also protein and proteomics. So we know that nucleic acid could be amplified, but you could amplify protein. Who knows, in the future you could. Nucleic acids show uniform behavior in purifying and handling, which means that they are actually easy to purify, and in terms of handling, it's not that hinky-panky, apart from probably RNA. But protein, no. Basically, you have to have a very specific kind of like procedure in terms of when you want to purify, and also in terms of handling, because it's actually more or less it requires specific techniques. So for nucleic acid, they are self-complementary because we know that usually they come in double-stranded, even though for RNA it doesn't have a double-stranded, but when you reverse translate it to cDNA, you have complementary. In protein, no, there is no complementarity. Nucleic acid have limited but increasingly appreciated modification, which means that it doesn't really have that much modification as compared to proteomics. Not to say that protein have limited modification, okay, no, meaning that basically to similarly answering to this, protein has a lot of modification occurring, especially post-translationally. And the last one is nucleic acids are stable to drying, spotting, etc. So that means that handling is actually at ease, more or less similar to this. But for protein, it's actually conditional because we know that protein is affected by heat. Now, what are the tools for proteomic study? So there are lots of tools, okay, but what we will see is actually some technologies for identifying protein. Okay, the one that I've actually highlighted and have put a star there, and also technologies for quantifying protein expression. So, and the last part is that we're going to take a look at bioinformatic tools. So we will look at some separation proteins and peptides technologies in terms of chromatography, followed by how you could assess the protein-protein interaction. Okay, this is actually coupled with this. Now, okay, let's have a look. So this is more or less how protein is being isolated from the beginning. So it depends on sample type, location of protein of interest, and also the required protein yield. So you really need to justify all this before you start your purification or isolation procedure. Secondly, propose downstream application. You also need to know what are you isolating the protein for. You couldn't just isolate it just for the fun, for the sake of it. Okay, so you need to know what downstream application are you going to be using it, because the purity, the required yield, matters. All right. So in terms of protein isolation procedure, there's a lot of method, but okay, the basic method is cell lysis. Okay, so whatever that the method is named or being called is basically based on cell lysis. So this is the best possible yield and purity for different species of organisms, sample types, and also for target molecule or subcellular structure. So this is done either using reagent-based method or physical method. Okay, so I have a blender here, but you could use multi-vessel and whatnot. Right, how do you separate protein and peptides? So we know that peptides are those polypeptides before a protein is in its 3D structure. So you could separate it via electrophoresis. So not your normal DNA electrophoresis. So it's still called electrophoresis, but we call it as SDS page. Okay, SDS polyacrylamide gel electrophoresis. So it could be either one-dimensional or two-dimensional or capillary. So the advantages are no other technique offers comparable resolution with simultaneous quantitative analysis. Modern stains deliver differential proteomics, so we have lots of modern stains, which we will see in a while. It is highly compatible with peptide mass fingerprinting, which we will see in a few weeks. So it is also basically a very simple technique, but it doesn't come with it does come with challenges, which means that again there is nothing all sweet about it. Okay, there are some challenges. So in terms of labor intensive or sometimes not high throughput, that's why you need to more or less come out with a very good method of isolation in the beginning. Membrane and also other recalcitrant proteins usually would be problematic. So you need to identify if your protein is a membrane protein, you need to have extra careful of how you could actually purify it correctly. The last one is actually associated with two-dimensional, which we will cover in a few weeks. The reproducibility and also the yield. Most of the time, it's actually the reproducibility. It's quite hard to justify. All right, so let's take a look at the electrophoresis part. So we know that when we run DNA, you get bands, right? Similarly, actually in protein, that is in 1D. What I'm showing you now is actually the 2D gel. For protein, for 1D gel, you get bands. 2D gel, you get spot. So the difference, is that the bands, from 1D SAS page, basically one band, could represent more than one protein. But for the 2D, such as this, one spot, for example here, represent one protein only. It's very specific. So as you can see over here, this is basically the explanation of this. So any of these spots could be anything, kind of like serum and whatnot, and a lot of it would be rubisco, which is the most abundant protein on Earth for every organism. So let's have a look at protein stain. So for DNA, the stain that you commonly use would be ethidium bromide, or now we have a safe stain, which is basically an equivalent to ethidium bromide but safer on you when you use it. So in terms of protein, we do have similar stains, just that the stains is actually not as dangerous as ethidium bromide, but it is essential, because it is a gel base. So you need to get a depiction of the proteomes, either by the bands or by the spots. So it is an important component of gel-based quantitative proteomics, and all stains usually show some protein-to-protein variability in detection efficiency because it depends on the sensitivity of the stains, which we'll see. And all staining or imaging techniques usually have limitations in linear response range, meaning that it depends on your gel documentation and probably would be worse if you were to capture it with your handphone only. Now, these are some of the common stains. So there are lots of other stains, so I'm just giving you the basics. We have standard Kumasi R250. We have colloidal Kumasi G250. So R and G do mean something. We'll come that to in a while. Silver staining, which is much more sensitive as compared to the Kumasi. We have fluorescent stains, whereby you need to view it under basically UV light, and also we have specialty stains. What are they? We'll have a look. So we'll start off with the Kumasi R250. So the sensitivity is around 300 nanogram per tile spot or band. So either when you use it on 1D or 2D, it is fully compatible with MS or mass spectrometry, which is actually important if you were to identify your protein later on. So it basically has a reasonable linear response, which means that getting your picture, documenting it would not be that much of a problem. So it is also cost effective because it's actually the cheapest, okay, to buy so far. So protocol is about 1 to 2 days, okay, because it also depends on your protein abundance as well. But okay, even though it has reasonable linear response, sometimes the blue spots are difficult to image, okay, because of the reflectance in terms of the scan, also white background, also maximum base of pixel depth. So actually this a link to a YouTube video on how the CuMES-CR is being used as a stain. So the R basically represent red. So that means that, okay, if you use CuMES-CR, when you run your gel, okay, when you view it, okay, you would see that a tint of redness in the bands or in the spots. So by red, it basically is around blue, kind of like blue indigo color, but you will see a tint of red hues. Now, CuMES G250 is actually of colloidal, so basically it has a higher sensitivity, okay, around 50 nanogram per tile spot or band, and it is recommended when limit of detection of MS work is basically anticipated, or when you want to have a very good identification from your MS. So it is fully compatible with MS, so it should be no problem there. Similarly, with the R250, it has a reasonable linear response. It is also cost effective, doesn't really cost that much, I think about a few, well, I would say ringgit, okay, not to say a few ringgit, okay, probably like around 20 to 50 up until 100, okay, a bit more than the R250. And basically it by right should have a 4-day protocol for maximum sensitivity, but most of the time we use similar protocol as in the R250. So again, the blue spots are still difficult to image. Alright, so the third stain is the silver stain, which is quite common as well because it has a high sensitivity, okay, around 1 to 5 nanogram per tile spot or band. So you can actually beautiful image. What we meant as beautiful is that not that the color is beautiful, because it doesn't really give you that color, because the color is kind of like dark brown or more or less kind of like a bit blackish. So it is good for pattern gold. So it is also cost effective because even the silver stain, okay, may be expensive, but you could use it for one bottle of about 25 gram, I think so. So probably could last you years. But okay, it is basically, okay, poorly compatible with mass spectrometry. So which means that if you were to identify it later on, don't stain it with silver stain because it is not compatible. You will not get anything identified. Trust me, I've done that. So some of the problem with silver staining is that it is too sensitive. Why? It could also cause to damage sensitivity because you need to be very careful when you expose it to the stain, okay, and when you develop it later on, because overexposure and overdeveloping, okay, would cause the background to be black. Hence, you won't be able to identify the band and also the spots. So for quantitation, it is not recommended, okay, because basically it will give a very false positive rough estimation, and it also has poor linear response. This is due to the sometimes overdeveloped exposure. But okay, it basically has a variable gel to gel sensitivity. It actually depends on how good you are at estimating how long to expose it and how long to develop it. Okay, so the hint is that most of the time we don't really use plastic, we use a kit, okay, or basically make sure that your silver nitrate is basically what we meant as age. Your silver nitrate is that when you make it, okay, so don't use it straight away. So use it like after about, I would say, leave it for about a month and then use it. Usually it's much better. Now, silver versus the CCB, which is the CuMES blue, okay, so if your goal is sensitive patterning of a proteome, silver is a good place to start. But again, you need to be very careful, okay, of the exposure and also developing time. But if your goal is preparative separation of unique protein for MS identification, so of course CuMES is a good place to start because it's also compatible with the MS. Now, the other two stains are fluorescent stains from the Cypro family, and of course different brands have different names. So Cypro is actually from the first brand that came out with it, okay. So sensitivity is similar to actually in silver stain, around 1 to 5 nanogram per tile spot or band. So it is referred to as Cypro Ruby Red, okay, which is actually less for other Cypro, which is ruby red is actually the best. We have red, we have green, we have yellow, and we have blue, I think, which is not a lot of people like to use the blue because they actually prefer to use chromacy if they want a blue color. So it has excellent linear response, so 3 orders of the magnitude, but it is not cost effective. So I put here 220 USD per liter. This was in 2016, I think, so the cost would have increased now. And the downside is that you require UV excitation. You cannot see it with your naked eyes, okay? So you have to have a UV excitation, okay, to view your gel. So usually you need a laser scanning or a UV transilluminator to view it. And of course, okay, to take a picture with your handphone alone is almost impossible. So it is also incompatible with manual spot cutting. This is for 2D work, okay, usually if you want to spot it and then later on try to quantify and whatnot. So it is compatible because usually a spot cutting machine, okay, sorry, if you use a spot cutting machine, okay, basically you could use a UV filter, but for manual cutting, which means that you won't be able to see it unless you were to do it on UV transilluminator, which is not recommended because 30-second hour of exposure without proper eye maintenance or shield, okay, would cause you blindness for about a week. So it's actually, of course, okay, not compatible. So apart from that, we also have specialty stains. So these are fluorescent as well. So usually what I have here is actually from Invitrogen, and I'm not trying to promote Invitrogen by anyway, but because it is the one that I've used before. So we have ProQ, okay, and the names varies for diamond, emerald, emerald 300, amber, and also oligohistidine gel stain. So the difference is basically in terms of for the PTM that you are aiming for your protein, okay, in the 2D gel. So difference in terms of the names, okay, is basically for the PTM that occurs to the protein, if you know about it. So usually this stain, because the name says specialty, so you need to have a very special reason, a very specific reason to use it. Okay, so if you know that there are membrane protein involved, okay, so you could use ProQ Amber. Right, so let's have a look, okay, at basically some of the staining effect. Okay, so this is sequential or composite images. So this is done, okay, using Cypro red, ruby red, okay, and also blue, okay, as you can see over here. So like I said, okay, usually people don't really prefer the blue, but because if you need a fluorescent, then well, you still have to use the blue. So it could have, okay, you could see basically over here, this is actually done. Okay, so this is actually from the Invitrogen website. So this is basically done all in one gel, okay, but using 2 different stains. So the 2 different proteomes basically have been labeled, okay, differently. Now, just so that you could see the differences among the 5 stains that I've said before, so I'm not showing you the fluorescence here because it is shown here. So this is basically between the R, the G, okay, silver, which is actually nice, but you need to be very careful because the background could be very black and sometimes you couldn't even differentiate between them, okay, between the 2 bands, okay. And also these are some of the fluorescent stains available. So now, as you can see, okay, when I was saying R, which means that, okay, red, you won't be able to see it here, but when you view the gel, okay, in front of you, you can actually see, okay, a tint of redness in it, okay, so making it look like kind of like purple. But when you use G, okay, your gel will look like blue because G is actually for green, so it gives out green hues, making it look kind of like bluish, okay. Alright, so apart from that, okay, we also have one kind of application or tools, okay, for proteomics, which is dye or difference gel electrophoresis. So it means that you have around, well, usually more than 1 proteome, okay, so you have 2 or 3 proteomes that you can actually mix around together, okay, so basically similar with the gel that I've shown you earlier on with the fluorescent stains. So proteome 1, okay, for example, over here is labeled with, okay, Psi3, which is the false pink, okay, but you see that it's actually a bit kind of like purplish. And second proteome, okay, is labeled with Psi5, which is a false green. So you mix it, you run it on a gel again, and then you scan it for the fluorescence. So what you see here, okay, is that if there is any similarities between the protein in terms of its expression, then you can see kind of like, if you see over here, there is basically an overlap here, and probably here is like, if I'm not wrong, okay. Now, so we know that protein separation and also peptide separations, okay, could be done further by using chromatography, which we will cover, I think, next week or the week after. So it is applicable to whole protein and also peptides, and it's actually simple linear method. But you need to do a cleanup in terms of C18, column 18, okay, or basically a specialized column of whole peptide population. So it further separates peptide from each other, not only just protein, okay. And apart from that, we also have a lot of different kinds of chromatography. So in theory, there are 4 to 5 basic chromatography, but okay, for different class, okay, basically lots of different methods to do the chromatography. So in terms of peptides, usually we use a mud pit technique. Okay, I will cover it later on. Okay, so I'm not going to discuss it now. And usually, for protein, okay, so the proteome works again at all is the one that we basically base it on. So that is one of the first literature that comes up. I actually forgot the date, so that's why I actually didn't put it there. But it was way in late 1990s. So apart from that, again, one of the chromatography, which is an affinity purification, whereby you could basically separate the protein or peptide based on affinity, is actually quite crucial, okay? Especially if you have protein family and also protein complexes, but you need to know what is the affinity of the protein or its complexes. Now, how do you purify large proteomes? So you could purify large proteomes, okay, but you need to have a very targeted approach on the purification method. So it could be a whole organism, organs, or tissue, especially for eukaryote or larger animals. You could also use laser microdissection or cell sorting, but laser microdissection would be very expensive. And it could also sometimes involve major organelles, which means cell walls and plasma membranes, nucleus, mitochondria, etc. And also sometimes it could involve fine organelles, but a combination of them, such as mTOR, again, nuclear pores, and so forth. Ooh, okay, so this is actually probably because I stretch it, so it's actually not very clear, okay, but this is basically some of the target method that you could use. So this is just a general overview, not definitely that you need to use it. So you first and foremost need to determine the purity of your protein, because it also depends on the downstream processes or application. So you can have a low, medium, or high quality, and the different kind of chromatography that you apply basically depends on the purity of it. So you could target, but in the end, when you see that, okay, let's say for example you target for a medium purity, but you get a low purity, so you basically have to suit the chromatography, the separation technique, okay, that is good for the kind of purity that you get. Okay, right. So basically that's it again for the proteomics, the introduction of it anyway. So next week we will move further again into the gist of the proteomics part of this course.